Bismillahirrahmanirrahim. Here's a case of newborn presenting with lethargy and refusal to feed. The accused in this scenario, which point towards the diagnosis of maple syrup urine disease, are burnt sugar smell in diapers, along with ketoacidosis and dietary restriction to prevent mental retardation. So, diagnosis is the maple syrup urine disease. Metabolic defect is the impaired carboxylation of branch chain amino acids. As we can see here that the branch chain alpha keto acid dehydrogenase that oxidatively decarboxylates the branch chain amino acids is defective, and there is accumulation of their keto acids and alpha hydroxy acids in the blood and urine. Alpha keto dehydrogenase complex is a multi-subunit enzyme, and that is alpha keto acid is decarboxylated to form a hydroxyethyl derivative bound to the reactive carbon of thymine pyrophosphate. Here the thymine pyrophosphate is the coenzyme of the alpha keto acid decarboxylase complex, or E1. Next, the hydroxyethyl intermediate is oxidized by the transfer to the disulfide form of the lipoic acid, which is covalently bound to the dihydrolipoyl transacetylase. So, this domain of the alpha keto acid dehydrogenase complex transfers acetyl group bound as a thioester to the side chain of the lipoic acid to the hydroxyethyl intermediate. The sulfhydryl form of the lipoic acid is oxidized to dihydrolipoyl, and the hydroxyethyl intermediate is now converted to the CoA form of the respective amino acid. The E3 domain regenerates the oxidized form of the lipoic acid. It is important to remember here that the E3 domain of this complex is identical to the E3 domain of the pyruvate dehydrogenase complex, which converts pyruvate to acetyl-CoA and is important in energy generation through TCA cycle. Now, the likely biochemical cause of the ketoacidosis is the accumulation of the ketone bodies, namely beta hydroxybutyrate, acetoacetate, and acetone. And not only that, the alpha keto acids formed after the transamination of the leucine, isoleucine, and valine, their accumulation also contributes to the aggravation of the alpha ketoacidosis. So now let's discuss the biochemical aspects of the ketoacidosis. The reason for ketone bodies formation is the energy deprivation. Normally, during these degradative reactions, NADH and FADH2 are generated for energy production. One NADH is produced during the oxidative decarboxylation of each of the branch chain amino acids. Besides, one FADH2 is produced when isovalyryl-CoA is converted to acetoacetate during degradation of leucine. One FADH2 and two NADH are produced when isobutyryl-CoA is converted to propionyl-CoA during degradation of valine, and one FADH2 is produced when isovalyryl-CoA is converted to HMG-CoA during degradation of leucine. If this process is impaired, the role of the branch chain amino acids as metabolic fuel will be lost, and body is going to face energy deprivation. Not only the energy generation from the branch chain amino acid catabolism is impaired, but also from glucose. Under normal circumstances, glucose through glycolysis forms two molecules of pyruvate which enter mitochondria and are converted to acetyl CoA. So, this step gives rise to the formation of 3 ATP per molecule of the pyruvate. Further, 10 ATP are generated for each molecule of acetyl CoA entering the TCA cycle. Since E3 domain of pyruvate dehydrogenase and alpha ketoacid dehydrogenase are identical, the functioning of pyruvate dehydrogenase complex may also be compromised, and acetyl CoA will not form from pyruvate. This may this may channelize the pyruvate towards the formation of lactate, further exacerbating the acidosis. As the body will be starved of energy, fat stores are likely to be mobilized, with acetyl CoA being formed from the beta oxidation of the fatty acids and subsequent formation of the ketone bodies, which are acids. And as we discussed earlier, that alpha keto acids are also going to accumulate because the pathway of the degradation is blocked, and the metabolites prior to block will accumulate in blood, and these are the alpha keto acids formed after the common transamination reaction. Here we can see that the alpha keto acids, alpha keto isocaproate, alpha keto isovalerate, and alpha keto beta methyl valerate have been synthesized before the block, so these will accumulate. Regarding the burnt sugar smell, sotalol is a chemical compound formed during abnormal metabolism of isoleucine and is responsible for the burnt sugar smell. Accumulation of leucine along with the alpha keto acids may cause mental retardation.